Welcome and thank you for standing by. All participants will be able to listen only until the question and answer session of today's conference. At that time, if you would like to ask a question, you may do so by pressing star, then 1, and recording your first and last name. Today's conference is being recorded. If you have any objections, you may disconnect at this time. I would now like to turn the call over to your host for today, Ms. Dacia Beard. Ma'am, you may begin.
Good afternoon. My name is Dacia Beard, and I'm a communications lead in the National Cancer Institute's Office of Communications and Public Liaison. Welcome to the Frontiers in Nutrition and Cancer Prevention online webinar series brought to you by the National Cancer Institute Division of Cancer Prevention Nutritional Science Research Group. To ensure uninterrupted streaming of the online portion of this presentation, please close any additional programs and windows on your desktop. If you have any technical difficulties during the presentation, please press star 0 for operator assistance or call the technical support number shown on the screen. As a reminder, this webinar is being recorded and will be archived on the NCI Division of Cancer Prevention's website. We will post the address at the end. Before we get started, please take a moment to familiarize yourself with the live meeting desktop. The question and answer box labeled Q&A is located on the top navigation bar. You can keep it open by clicking on it and then dragging the box to the right side of your screen. You may type your questions here at any time during the presentation, and the speakers will answer them during the Q&A period after the presentation, as time permits. Now I would like to introduce your moderator for today's webinar, Dr. Gabriela Rasuda, Program Director in the Nutritional Science Research Group, Division of Cancer Prevention at the National Cancer Institute.
Good afternoon. I would like to welcome you all to the 6th presentation of the Frontiers in Nutrition and Cancer Prevention online series. I am very pleased to introduce today's webinar, The Impact of Obesity on Cancer Risk. Before we get started, I want to thank Dr. Barry Kramer, our Division Director, and to my colleagues on the Steering Committee and Planning Committee who made this webinar possible. We are very excited that we have so many people joining the webinar today. We have researchers, physicians, nurses, registered dietitians and nutritionists, clinicians, fellows, and students with us from all 50 states of the United States and many countries all over the world, like Canada, Italy, Brazil, Sweden, and Argentina, just to name a few. I suspect that we'll hear a variety of interests and perspectives shared during our question and answer portions of the webinar. According to the U.S. Center for Disease Control and Prevention, more than 1/3, like 24.9% or 78 million of adults in the United States are obese. Growing obesity incidence is associated with detrimental health consequences, including cancer. In our webinar today, experts in the field of nutrition and cancer will evaluate the strengths and weaknesses of epidemiological data and the underlying molecular mechanisms and common pathways of obesity and cancer. During the presentations, gap in knowledge and new tactics to advance obesity cancer research will be discussed. Now, I would like to turn over the program to our first presenter, Dr. Edward Giovannucci. Dr. Giovannucci is a professor of epidemiology and nutrition at Harvard Chan School of Public Health. He is an associate professor of medicine at Harvard Medical School, and he is also an associate epidemiologist in the Department of Medicine of the Brigham and Women's Hospital. The title of Dr. Giovannucci's presentation today is Obesity and Cancer, Epidemiological Observations. When finished, Dr. Giovannucci will take a few minutes to answer questions. Dr. Giovannucci, please.
Thank you, Dr. Riscuda. It's a pleasure for me to be talking about obesity and cancer. I'm going to focus on the epidemiologic observations. As you all know, obesity has been associated with an increased risk of cancer incidence and especially mortality for many cancers. The American Institute for Cancer Research estimates greater than 100,000 cancers per year in the United States are attributable to excess body weight. Now, it's amazing how many cancers are related to obesity. There are not many risk factors that are cross-cutting across so many cancers. This slide shows the 13 cancers that have been associated with obesity. They range from GI cancers, endocrine, genitourinary system, quite a broad spectrum. I'm going to focus on the epidemiologic data. And as you know, in the epidemiology, we have to assess obesity or adiposity in a large population, so we have to use practical measures. And the most common is, body mass index, which is kilograms per meter squared, which takes into account a person's weight and their height. And you probably know these categories. Overweight is considered greater than 25, obesity greater than 30, and greater than 35 is even more severe obesity. Now, using this measure, a large study from the American Cancer Society estimated that in non-smoking individuals in the United States, about 14% of cancers are attributable to having a body mass index greater than 25 for men and about 20% for women. So at least in theory, this means that if we got everybody the body mass index below 25, we would prevent about 14% of cancers in men and 20% in women. So if we look at all the individual types of cancer, each cancer is different, but the proportion attributable to overweight is approximately 10 to 20%, as we've seen. Now, for two cancers, endometrial cancer and esophageal cancer, obesity is much more strongly associated, and the proportion attributable to BMI greater than 25 is greater than 50%. So for these two cancers, obesity is a very strong risk factor. But I'd like to point out that the percent of cancers attributable to excess adiposity is likely underestimated because most studies have used a single measure and a BMI cut point of greater than 25. So what happens if we use a stricter cut point of 22.5 kilograms per meter squared? So I just show one slide here this is from the health professional study. And I just want to make the point here that the relation between body mass index and cancer is linear. We use practical cut points like 25 and 30, but the association you can see is across the whole spectrum. In fact, you can see that using the bottom group, BMI 20 to 22.5 as a reference, there's a statistically significant 50% increase even in individuals with BMIs from 22.6 to 25. So even in the moderate range or in the lean range, not considered overweight, there is a relationship between adiposity or BMI and risk of colon cancer. So if in the American Cancer Society study, they concluded that 10% of colon cancers were attributable to BMI greater than 25. In our study, health professionals follow-up study, we use a stricter cut point of 22.5, and we use also multiple measures, so we try to get more consistent BMI measures over time, and we found that 30% of colon cancers are attributable to having a BMI greater than 22.5. Of course, this may not be, getting people below 22.5 may not necessarily be practical on a population basis, but at least it's important to know that the relationship is even stronger if you consider lower BMIs. Now, BMI is a useful measure in large epidemiologic studies, but it does have some limitations. And the two most important are it does not distinguish lean mass from adiposity, and it does not distinguish visceral from subcutaneous adiposity. So circumference measures are useful also. In this study, in the health professional study, we looked at waist circumference in colon cancer, and we actually found a strong association even after adjusting for body mass index. So it seems that waist circumference gives independent information than just body mass index. In this study, in the nurses' health study, they stratified body mass index, you can see, into three categories, and also then stratified by a large and lower waist-to-hip ratio. So if you can see with the reference group, with individuals less than 25 BMI less than 25, and also having the low waist-to-hip ratio, if you have BMI 25 to 29.9, which is the overweight, you see that those with the lower waist-to-hip ratio had a relative risk of 1.25, but it was actually 2.05, much stronger if they had a high waist-to-hip ratio. So even in the overweight range, the waist-to-hip ratio adds a lot of information. Now, visceral adiposity is important to consider. What is visceral adiposity? Well, circumference measures are useful in distinguishing adipose mass from lean mass. However, circumference measures, even waist circumference, does not directly assess visceral adiposity, especially among those with a relatively small waist circumference. I think this slide illustrates the point very nicely. This is from CAT scans from a published paper from Thomas. And as you can see in the CAT scan, this is taken at the level of the umbilicus. So these are 8 separate men, all have this exact same waist circumference. But you can see from going from the top left, you see much less fat. The adipose comes out as white, on the CAT scan. So you see the top left man has a visceral fat estimated at 0.5 liters. If you go to the bottom right, the visceral fat is 4.3 liters, which is 8 to 9 times higher visceral fat, even among men that have exactly the same waist circumference. So there's a lot of information that's not captured by even circumference measures. And there's studies, it's hard to use CAT scans or MRIs to estimate visceral adipose tissue in epidemiologic studies because they're costly. There's been studies done for colorectal adenoma, which is a precursor to cancer, because these are very common, and you only need to, measure about 1,000 to get good power. And we actually did a pooled analysis of the published studies, and we found that independent of BMI, you see a nice linear association between the visceral adipose tissue, or VAT, and the odds ratio of colorectal adenoma, with almost a 3-fold increased risk going to the high level compared to the low. Now, these measures, BMI, circumference, even visceral adiposity tissue, are static measures. They don't take into account changes. Now, ideally, we would like to do a large randomized trial to show that changes in weight, preventing weight gain or inducing weight loss, will reduce risk of cancer. But those studies are very hard to do for practical reasons. There is a little bit of epidemiologic studies, observational data that inform on this. So if we assume that adiposity is causally associated with cancer risk, two important questions are, would preventing adult weight gain lower one's cancer risk? And the second is, would intentional weight loss lower one's cancer risk? Let's look at the first question. And there are some epidemiologic studies that suggest this, and that this is preventing weight gain in adulthood would potentially lower risk. And this is a pooled analysis we did on studies of postmenopausal breast cancer, which has been associated with obesity, and we can see that adult weight gain strongly correlates with risk of breast cancer, and this is among women non-hormone replacement therapy users. So this at least is observational data that women who gained the most weight had the highest risk of breast cancer. And there are a few other studies. There's a study, for example, in colon cancer that also shows this pattern, but it needs to be looked at for other cancers. Now, there's another question is whether intentional weight loss would lower one's cancer risk. That's actually a much harder question to answer from an epidemiologic perspective. For three reasons that I list here. First of all, intentional adult weight loss is rare, so it's hard to actually get enough numbers. And then the second thing is much of the weight loss is not maintained over time. And third is that most of adult weight loss, if you look broadly at the population, is actually due to illness, underlying disease, and loss of lean body mass. So that may not be really what you're looking for. It's not intentional. There have been a few studies, and this is in a nurses' health study, in which they try to take into account some of these issues by looking at longer-term weight loss. And they actually did find a reduction. So women who lost weight, 10 kilograms, for example, or more over the menopause period, actually had about half the risk of developing postmenopausal breast cancer. The other sort of evidence is from studies of bariatric surgery, where people have surgery and then have a weight loss, and these are very obese individuals. These aren't perfect randomized trials by any means. They usually need to have a control group of women or men of similar weight that did not have the surgery. And as you can see in the slide here, at least for women, after the surgery, there was a pretty large reduction, primarily in breast and endometrial cancer, compared to what their rates would be expected. So this is a little bit of evidence that weight loss actually would impact your risk of cancer. Now, other speakers, particularly Dr. Dannenberg, is going to talk a lot more on mechanisms. So I just want to touch briefly, based not so much on detailed mechanisms, but more some of what the epidemiology suggests. And there are multiple mechanisms where obesity may be associated with cancer risk. And what's been most interesting in the past 10 years or so have been large studies that have linked pre-diagnostic blood levels of hormones such as insulin, insulin-like growth factor 1 or IGF-1, and estrogen. These are all linked to obesity and with various cancers. So, for example, here we can see that studies have clearly shown, multiple studies, that IGF-1 and estrogen is associated with increased risk of breast cancer. So for the 5 cancers listed, there is pretty compelling data that suggests at least there's an association between these markers and risk of cancer. And these blood samples are taken years, sometimes decades before the risk, before the cancer occurs. And just to show you one little bit of this, this is body mass index in cross-sectionally in the nurses' health study. You can see there's a strong association between BMI and bioavailable estrogen levels, almost a 3-fold, 3 to 4-fold difference between the low and the high group. And this corresponds almost perfectly with the risk of endometrial cancer, which is strongly associated with estrogen. And if you look at postmenopausal breast cancer, you also see that adult weight gain, interestingly, strongly predicts cancer in non-hormone replacement therapy users, women not receiving estrogen postmenopausally. But you see no association in estrogen users, which indirectly, not definitively, but indirectly supports the role of estrogen in breast cancer, because the women taking hormones already have high estrogen levels and are at high risk. but among the women not taking estrogen therapy, there's a nice correlation between weight gain, which is associated with estrogen, and their risk. I'll also point out that there are other mechanisms and that may be very specific for cancers, and I think local inflammation is an interesting concept here. For example, obesity is associated with acid reflux, which causes chronic low-grade inflammation in the esophagus. Barrett's esophagus, a precursor, and leads to adenocarcinoma of the esophagus. Gallstones are strongly associated with obesity, and probably chronic gallstones increased risk of gallbladder cancer through chronic inflammation. And fatty liver is very strongly associated with obesity and sometimes leads to inflammation and to liver cancer. So now I'm finishing up here, but I just want to note an important observation. Obesity may influence prognosis from cancer and cancer mortality beyond effects on cancer incidence. All that I've talked about so far has been on the incidence. But there are potential reasons why obesity may impact cancer mortality independently. It may, for example, cause more aggressive subtypes of cancer, or it may influence things such as diagnosis and treatment, which may make cancers, obese people have a poor survival. I just wanted to show these data. So for breast cancer, if you look at these two age groups, you look at the relative risk for a 10-unit increment in BMI, and you see a much stronger increment in risk of mortality than incidence. So for example, in the women 60 to 64 years old in this study, this increase in BMI was associated with 22% increased risk of getting breast cancer, but a 75% risk of getting and dying of breast cancer. So the impact of obesity goes beyond just incidence. And this study just shows recurrence. As you can see, the lowest risk is a BMI of 22, and then it goes up with increasing BMI. This is recurrence in women with localized breast cancer diagnosis, strong association. And prostate cancer is one cancer that's interesting because obesity is not associated with incidence, but it does seem to increase mortality. And obesity has been associated with higher grade prostate cancer, more advanced stage, and poorer survival. So just in summing up here quickly, we've seen that excess body weight increases risk of at least 13 types of cancers, broad spectrum of cancers. Cancer mortality may be increased independently of incidence. The effect of obesity on mortality may be even independent. There may be independent mechanisms that increase mortality beyond just increasing risk. Multiple mechanisms are involved, and Dr. Annenberg will talk about some of those. And the final note is the impact of obesity on cancer incidence and mortality may now be comparable to that of tobacco in the United States. As tobacco has gone down, still, of course, a very important risk factor, but as tobacco use has gone down and obesity has gone up, obesity is now becoming comparable to tobacco use as a risk factor for cancer. Thank you, and I'd be glad to take questions at this point.
Thank you. At this time, we'd like to begin the question and answer session of the conference. If you would like to ask a question at this time, please press star, then 1. You must record your first and last name to ask your question. To withdraw your question, you may press star, then 2. Once again, if you would like to ask a question at this time, please press star, then 1, and record your first and last name. One moment for the first question, please.
Yes, it's Gabriela Discuta. We actually received some questions through the chat box, and I will read some questions for Dr. Giovannucci. Dr. Giovannucci, thank you so much for great presentation. And the first question that I would like to read for you is any hypothesis or do you have any thoughts why the risk of some cancer is higher than others in relation to the BMI level? So I guess the question is, why not all cancers are showing the same relation to the obesity in regard to the BMI level?
Yes, I mean, that's a good question. Every cancer has its own set of risk factors, and, you know, for example, smoking is a strong risk factor for some cancers, not a risk factor for others, and actually even a little bit protective for some cancers. So there's you know, cancers are very complex, and it's hard to think that there would be a factor that would be strongly related similarly to all cancers. Now, some cancers, like, for example, endometrial and breast cancer, they're tissues that are strongly related to estrogen. Estrogen is a strong proliferative component of these tissues. So it's not surprising to me, for example, that in postmenopausal women, most of the estrogens are made from adipose tissue. So for these cancers, you would almost predict that there would be a strong association between obesity and cancer. For other cancers, you know, they may not have as clear hormonal factors that are associated with obesity. So I think it's hard to say, you know, when we go down the list, we don't have time for each cancer site, but I think for some cancers, the hormonal link is much stronger than for others, and that's the main reason.
Thank you so much, Dr. Giovannucci, and I have one more question for you. I mean, there are more questions, but I will read one more due to the time limit. So this question is your impression that the visceral adiposity is as important, more important, or same important as BMI level in regards to the cancer risk? Because I think you presented, I'm just adding my comment, that you presented the visceral adiposity as being an important factor related to cancer risk. So how would you relate the visceral adiposity and the BMI level? Which one is more important than other? Do we have enough data to make any thoughts about?
Well, yeah, I mean, the visceral adiposity, some people believe it's the most important component of adiposity that's related to metabolic aberrations such as insulin resistance. Now I think the main point to take from some of the slides that I showed is that when you get, like, for example, individuals with a very high BMI of, let's say, 35, for example, those individuals very likely have a lot of visceral adiposity. It's hard to get a BMI that high without having a lot of visceral adiposity. But when you get to lower BMIs, as I showed in that slide from CAT scans, you can see even men with seemingly normal waist have a wide range of visceral adiposity. So when you get into the normal range of BMI, that's where the visceral adiposity, I think, becomes much more predictive of risk. So I think they're both two ways of getting at the same thing. But I think it's maybe one way to think of it is the visceral adiposity is more useful at the lower end of BMI, where you don't really, you might say a BMI of 25 is normal, but that could be very different from an athlete, for example. A marathon runner can have a BMI of 25, or a couch potato can have a BMI of 25. So I think that's where visceral adiposity might be more informative.
So you're making the difference between people having the same actually weight, but a different waist or visceral adiposity, so they would be placed at maybe higher risk of
yeah, that's right.
Even being in the normal BMI level.
That's right. Yeah, BMI I think is more informative at the upper levels. So people with BMIs above 30 are almost clearly always obese, unless they're super athletes or something. But at the lower end of BMI, individuals can still have a wide range of adiposity, especially visceral adiposity, but that's not really captured by the BMI. So it's sort of a limitation of the measure of BMI. You can do it that way.
Okay, thank you so much, Dr. Giovannucci. And for additional questions, we will provide your contact information and our contact information to follow up with the emails in case the participants would like to continue this. And we need to move to our next presenter, Dr. Andrew Dannenberg. And Dr. Dannenberg is the Associate Director of Cancer Prevention at the Sandra and Edward Meyer Cancer Center, and Henry Earl, MD, Roberts Family Professor of Medicine at Weill Cornell Medical College of Cornell University, where he is also Professor of Medicine in Surgery, Weill Cornell Medical College. Dr. Dannenberg will discuss the obesity-inflammation connection, implications for breast cancer. When Dr. Dannenberg will finish, we'll have time for a few questions. Dr. Dannenberg?
Thank you very much for the opportunity to speak. As shown in this slide, the title of my presentation is the Obesity-Inflammation Connection, Implications for Breast Cancer. Obesity is a risk factor for the development of hormone receptor-positive breast cancer in postmenopausal women. It's also, as you've just heard, a poor prognostic factor for breast cancer patients, regardless of menopause or hormone receptor status. There are a variety of pathways which have been suggested to be important in explaining the link between obesity and breast cancer. For example, insulin resistance can increase the risk of breast cancer and is commonly found in association with obesity. Both high levels of insulin and elevated levels of free IGF-1 have been suggested to be causally linked to the pathogenesis of breast cancer. In the obese, one sees, as you've heard from Dr. Giovannucci, increased estrogen synthesis. This reflects both an increase in adipose mass, adipose tissue being an excellent source for estrogen synthesis. Additionally, several years ago, we reported that levels of aromatase, the rate-limiting enzyme for estrogen biosynthesis, were increased in association with elevated BMI. Additionally, one finds a decrease in SHBG, steroid hormone binding globulin, in the obese, and consequently, there's an increase in free estradiol. And it's the free estradiol which we believe is important in the pathogenesis of breast cancer. In the obese, one also finds altered adipokine and cytokine production. From the standpoint of adipokines, one sees elevated leptin and reduced levels of adiponectin commonly in the obese, and both elevated leptin and reduced levels of adiponectin have been suggested to play a role in breast cancer. It is also common to have a low-grade systemic inflammatory syndrome in the obese, and macrophage-derived pro-inflammatory mediators, including TNF, IL-1 beta, and interleukin-6, have all been suggested to play a role in obesity-related breast cancer. During the remainder of the presentation, I am going to focus on inflammation, which is associated with many of these pathways that I've just mentioned. In this slide, one sees a cartoon demonstrating that obesity causes an inflammatory state. It's been known since approximately 2003 that obesity is associated with adipose inflammation. In the obese, one sees adipocyte hypertrophy. Ultimately, presumably due to hypoxia, or at least partially due to hypoxia, a large cell will die, depicted by this brownish cell that I'm pointing to. In the context of an adipocyte becoming sick or dying, macrophages are recruited. These are depicted by these star-like cells, and they form a halo or envelope around the dead or dying adipocyte, and ultimately phagocytose that dead adipocyte and become foam cells in that process. One commonly sees increases in levels of pro-inflammatory cytokines in association with crown-like structures. Crown-like structure simply refers to this crown of macrophages decorating the dead or dying adipocyte. I will use the term crown-like structure, or CLS, during the remainder of the presentation. Finally, I should add that in the context of obesity, one sees increased lipolysis and elevated levels of free fatty acids, specifically saturated fatty acids, have been reported to activate macrophages, contributing to the pro-inflammatory state. Our next objective, therefore, was to determine if crown-like structures of the breast, or CLS-B, exist in women and correlate with body mass index. Many of you will probably be aware that there is a link between chronic inflammation, as Dr. Giovannucci just mentioned, and increased risk of a variety of cancers, but at the time that we did this work, there was no known chronic inflammatory state in the human breast. So, our initial study design was to evaluate breast tissue from 30 women who underwent mastectomy and to carry out routine H&E staining and CD68 immunohistochemistry looking for crown-like structures. CD68 stains specifically for macrophages, and it's much easier to see a macrophage when it's stained by CD68 than by simple H&E. This is what we found. Crown-like structures of the breast are in fact common in overweight and obese women. Here you can see a crown-like structure based on H&E, dead or dying adipocytes surrounded by a halo of leukocytes, which prove to be macrophages, and here by CD68 staining, you can see the macrophages, a crown-like structure of the breast, or CLS-B. And when we broke the code, as shown at the bottom, you can see that these lesions were much more common in the overweight and obese than in the normal. To date, we've evaluated several hundred samples, and we find crown-like structures in 85 to 90% of breast samples from obese women, a little bit more than half of overweight women, and fully a third of normal-sized women, and I will say more about the normal-sized women later on during the presentation. In this slide, we demonstrate that increasing body mass index is associated with increased breast inflammation. On the x-axis, you can see body mass index, and on the y-axis, we scored the severity of inflammation, and you can see overall as BMI increases, so does the severity of breast inflammation. However, there are outliers. Here you can see 3 women who were overweight and did not have breast inflammation, and here's a normal-sized woman who did have breast inflammation. So the take-home message is that overall one sees a relationship between elevated BMI or between BMI and the severity of breast inflammation, but for normal-sized women and overweight women, you can't make a prediction simply looking at body habitus. You can easily be fooled. A normal-sized woman could have occult breast inflammation. An overweight woman might or might not. So armed with this information, our next question was to determine whether or not the presence of crown-like structures had clinical significance. So our goal was to determine whether breast white adipose tissue inflammation manifested as CLS-B is associated with short and recurrence-free survival in women who develop metastatic breast cancer. Shown here are the clinical pathological characteristics in patients with recurrent breast cancer. So as you can see at the top, there were 127 women in this retrospective study. 52 of the 127 women had crown-like structures of the breast. 75 of these women were crown-like structure negative. And let's focus for a moment on those who are crown-like structure positive. You'll note, on average, compared to the crown-like structure negative women, they were older. They were more likely to have elevated BMI. Crown-like structures of the breast or white adipose tissue inflammation of the breast was associated with the postmenopausal state. And interestingly, features of the metabolic syndrome, including hyperlipidemia, hypertension, type 2 diabetes, were more common in association with crown-like structures of the breast. That's relevant because metabolic syndrome has been associated with an increased risk of breast cancer in some studies and also worse prognosis. So our data shown here strongly suggest that breast adipose tissue inflammation associates with features of the metabolic syndrome. Next, we went on to evaluate distant recurrence-free survival. Shown at the top is a graph whereby solid line represents CLS-B negative women, Again, crown-like structures measured at the time of index mastectomy, and the dashed red line represents those who had white adipose tissue inflammation or crown-like structures of the breast at the time of index mastectomy. And you can see quite clearly that the time to recurrence was earlier in the presence of crown-like structures of the breast or white adipose tissue inflammation. Shown below, in numerical form, the median time to recurrence was 20 months in those who had white adipose inflammation at the time of index mastectomy, and it was 26 months in those who had no evidence of inflammation at the time of mastectomy. By univariate analysis, this difference was statistically significant. When we adjusted for other factors that could have predisposed to recurrence, the presence of CLS-B or crown-like structures of the breast or white adipose tissue inflammation in the breast remained associated with an earlier time to recurrence. So based on these observations, we conclude that breast white adipose tissue inflammation is associated with metabolic syndrome. In patients who develop metastasis, white adipose tissue inflammation is associated with shortened distant recurrence-free survival. And so the question becomes, does white adipose tissue inflammation promote the progression of breast cancer via local effects, systemic mechanisms such as hyperinsulinemia, or both? So to address this question, we carried out the next study. The goal was to determine whether white adipose tissue inflammation is associated with local or systemic effects that can potentially promote breast cancer. This study involved assessing CLS-B status in 100 women, all comers regardless of BMI. We then carried out RNA-seq on non-tumorous breast tissue to evaluate local effects and to evaluate potential systemic effects. Fasting serum and plasma were used to perform lipid profiling, to look at hsCRP and IL-6, markers of systemic inflammation, and insulin and glucose under fasting conditions, and we also did metabolic profiling, small molecule profiling. And here is a description of these 100 women. In this particular study, roughly half of the women that were enrolled had crown-like structures of the breast. Once again, they tended to be a little bit older. Again, the presence of CLS-B was associated with elevated body mass index. They were more likely to be postmenopausal if CLS-B positive compared to CLS-B negative. And as you can see, there was a significant increase in the incidence of a clinical diagnosis of dyslipidemia in those who were CLS-B positive compared to those who were CLS-B negative. Here one sees a heat map representing the RNA-seq analysis. And breast white adipose tissue inflammation, as defined by the presence of CLS-B, is associated with numerous changes in gene expression. So shown at the bottom, you can see the CLS-B positive cases versus the CLS-B negative cases, and this white line divides the two groups. Each column represents a different human subject, each row represents a different gene, and these represent the 50 most upregulated genes, red being increased, green being low, and the 50 most decreased genes in association with CLS-B positivity. So the take-home message is that this histologic finding, crown-like structures of the breast, a histologic biomarker of inflammation, is associated with significant changes in gene expression in the breast, which could contribute to the development and or progression of breast cancer. But what about systemic effects? In fact, breast white adipose tissue inflammation is associated with biomarkers of cardiovascular risk. Here we see a nearly statistically significant association between CLS-B positivity and elevated levels of LDL cholesterol, statistically significant reduction in HDL cholesterol, the CLS-B positive versus CLS-B negative women, and elevated levels of triglycerides in CLS-B positive versus CLS-B negative subjects. Breast white adipose tissue inflammation is also associated with elevated hsCRP, IL-6, fasting insulin and glucose. Panel A, you can see the hsCRP levels. In B, the IL-6 levels being increased in the CLS-B positive versus the CLS-B negative. and one also sees, as already mentioned, elevated levels of fasting insulin, fasting glucose in the CLS-B positive cases. Now, what I'm not showing in the interest of time is the fact that breast adipose inflammation is a sentinel for what is a more diffuse inflammatory process involving other adipose depots. And once you know that, it makes good sense that there might be a blood signature in association with a systemic low-grade inflammatory disorder. Here are the results of our mass spectrometry-based metabolomic profiling study. Again, a heat map. We were able to demonstrate that breast white adipose tissue inflammation is associated with altered levels of more than 50 plasma metabolites. Again, red represents high levels and green represents low levels. Each column, again, represents a different human subject, and each row, in this case, represents a different small molecule. And if you focus your attention on the two groups on the right, you can see the overweight and obese, OW and OB, were either CLS-B negative or CLS-B positive, and I think you can see very clearly a large number of metabolites were increased in association with white adipose tissue inflammation, and a smaller number shown at the top decreased in association with breast white adipose tissue inflammation. Now, this is perhaps the most important slide of the presentation. We broke the code, and here you see that breast white adipose tissue inflammation occurs not just in the obese, and in this particular study it was virtually all of the obese, but also in roughly a third of the normal-sized women. So that implies that a third of women with normal BMI who are visiting their physicians have occult breast inflammation. And a significant medical dilemma to this day relates to why do normal-sized women who are not BRCA1 or BRCA2 mutant carriers develop breast cancer? So could occult breast inflammation be causally linked? So we went on and carried out RNA-seq on these normal-sized women, and here one sees the results. The take-home message is that breast white adipose tissue inflammation is associated with numerous changes in the transcriptome in normal BMI women, and at the bottom you can see groups that say yes. Yes refers to being CLS positive, no CLS negative. So the take-home message and the key point really is that occult breast inflammation in normal BMI women, normal-sized women, is associated with changes in gene expression, local changes in the breast. Now, one of the changes that was most striking and perhaps most meaningful from my perspective was a change in aromatase. Aromatase is the rate-limiting enzyme for estrogen biosynthesis. There are several FDA-approved aromatase inhibitors. Aromatase inhibitors are not only useful for the treatment of hormone receptor-positive breast cancer, but they have been shown in very, very large trials to lead to about a 50% reduction in the risk of hormone receptor-positive breast cancer in high-risk women. So what did we find in our normal-sized women? We found that breast white adipose tissue inflammation is associated with elevated levels of aromatase in normal BMI women. So shown here are the CLS-B positive normal-sized women versus the CLS-B negative normal-sized women, and we found about a one-fold increase in the level of aromatase in those normal-sized women with occult breast inflammation. It is intuitively reasonable to anticipate that if a woman is walking around long-term with elevated levels of aromatase, that this will increase the risk of breast cancer. Of course, that remains to be proven, but certainly it is intuitively reasonable. We went on and were able to demonstrate that breast white adipose tissue inflammation, in normal BMI women is associated with insulin resistance. So here you see the CLS-B positive normal-sized women versus the CLS-B negative normal-sized women, and you can see a nearly statistically significant increase in insulin, statistically significant increase in fasting glucose. HOMA2IR is a way of calculating insulin resistance. So there was relative insulin resistance in the normal-sized women who had occult breast inflammation. Now, why is this important? Well, as mentioned earlier on, hyperinsulinemia has been shown previously, if one considers all comers, to be associated with increased risk of breast cancer. But in 2015, in work from a different group, not from our group, it was shown that elevated insulin levels are associated with increased breast cancer risk in normal weight women. So in this case, metabolic unhealth versus metabolic health is defined by insulin levels, and one sees about a doubling in the risk of breast cancer in those who were defined as metabolically unhealthy because they were hyperinsulinemic. So I would submit to you, based on our recent discovery that occult breast inflammation is associated with hyperinsulinemia, that adipose inflammation is likely to contribute to elevated levels of insulin and increased risk of breast cancer, even among normal-sized women. So, based on these findings, we conclude white adipose tissue inflammation is associated with numerous changes in gene expression in the breast. White adipose tissue inflammation is associated with systemic changes that occur in the metabolic syndrome. White adipose tissue and its associated pathophysiology occur in a meaningful subset of normal-sized women. White adipose tissue inflammation is a potentially targetable process linking obesity to breast cancer. So, in the last few minutes, I'd like to briefly discuss our nascent efforts to reverse this process. It is simply not good enough to identify a new disease process. Obviously, we need to be able to diagnose it, and we need to be able to treat it. So from the standpoint of potential strategies to reverse this process, we are considering lifestyle changes, such as exercise, changes in diet. Will saturated fat matter, for example? Or might pharmacologic interventions be developed to disrupt the obesity-inflammation axis, thereby reduce the risk of breast cancer or improve prognosis? So these are data that we have generated using mouse models. And let me walk you through these data. It represents one preclinical strategy which may in fact translate to humans, to women, but this remains to be proven. We studied caloric restriction, and we were able to demonstrate that caloric restriction attenuates high-fat diet-induced weight gain. So in this particular mouse experiment, the green line represents lean mice, low-fat, ovary-intact mice. The other groups, the mice were overectomized to mimic the postmenopausal state. Remember early on I said obesity is a risk factor for breast cancer in the postmenopausal state. So to mimic that, we overectomized mice and then put them on a high-fat diet. We fed the high-fat diet to induce obesity in an inflammatory state, and then we randomized the mice to three groups, represented by the blue line, those that could continue eating ad lib, and as you see, they gained weight. Or we subjected two additional groups of mice to 30% caloric restriction for either 7 or 14 weeks. Same bad high-fat diet, but 30% calorie restriction for 7 or 14 weeks, which you can see did not induce weight loss, but protected against weight gain. And this is what we found. Here you see that caloric restriction reverses obesity-induced mammary gland inflammation. So panel A represents an H&E stain of a crown-like structure in the mouse mammary gland. And in panel B, we quantified the number of crown-like structures in the mammary glands in the different groups. And you can see that in the lean, low-fat, ovary-intact mice, there was no detectable inflammation. But by overectomizing a mouse and feeding it a high-fat diet to cause substantial weight gain, we induced inflammation in the mouse mammary gland. If we subjected the mouse to 7 or 14 weeks of caloric restriction, 30% caloric restriction that is, we dramatically reversed occult inflammation in the mouse mammary gland. I can also tell you, because it's encouraging, we've gone as low as 10% calorie restriction and also seen substantial benefit in the mouse. Here we went on to carry out molecular studies. We like to complement histologic analyses with molecular studies. Here we looked at NF-kappa-B binding activity in the mammary gland. NF-kappa-B is a transcription factor which is implicated in both pathogenesis of inflammation, cancer, and inflammation-related cancer. And here you can see that in the obese mice, The high-fat oophorectomized mice, one sees a marked increase in NF-kappa B binding activity compared to the lean mice, and you can see that either 7 or 14 weeks of calorie restriction led to a marked reduction in NF-kappa B binding activity consistent with the histologic findings which I shared with you. And finally, I think we need to be open-minded when we think about trying to translate these findings to humans. These are the interventions we are considering. Weight loss, yes. We're very interested in hormone replacement therapy. Exercise needs to be evaluated. We are beginning preclinical studies to look at potential medications that might reverse this. And, of course, combination regimens may be most prudent. And all of this needs to be personalized and tailored to the individual. And so it could be that in some individuals, a little bit of weight loss and medication may prove to be important, where in somebody else, weight loss alone may be practical. On that note, I thank you for your attention, and I am here to address any questions. Thank you.
Thank you so much, Dr. Dannenberg, for a great presentation. Indeed, we have a few questions for you, and I'm reading the first question. If was any study conducted in women with BMI equal or lower than 25 to evaluate their visceral adiposity and to see if there is any correlation between visceral adiposity and the inflammatory status?
That's a great question. The answer is no. I can say that with confidence because we were the first to start evaluating breast inflammation. But I can also tell you that breast inflammation is likely to correlate with altered visceral adiposity. We have new data to show that the adipocyte size is larger in those who have a normal BMI who are inflamed versus those who are not inflamed. And later this month, we will, in fact, initiate a study to look at body composition versus breast inflammation, including but not limited to normal-sized women.
Okay, thank you. Yes, so it appears that it is a correlation, maybe. And another question we have is, has there been any research on the association of decreased physical activity in the normal BMI women that would be related to the increased inflammatory status due to the more sedentary behavior?
Another great question. We've just conducted our first retrospective study to evaluate physical activity, and the data haven't been fully analyzed. The study is too small, but we, too, fully agree that it will be very important to evaluate the role of physical activity, exercise, as it relates to adipose inflammation, not only in the normal-sized individual, but also in the overweight and obese. So needs to be done, hasn't been done adequately to date.
Okay, thank you, Dr. Dannenberg. Only one more question because of the time constraint. So what would you think about measuring insulin level as a possible factor in evaluating the breast cancer risk? So basically in women having BMI equal or lower than 25?
So that is also an excellent question. I think insulin will provide some insight, but not adequate insight. In other words, although we find a relationship between hyperinsulinemia and inflammation in the normal-sized woman, there is plenty of overlap between the inflamed and the uninflamed. Also, single-point measurements can have their own limitations. So we're more interested in the possibility that a series of blood biomarkers may prove useful for risk assessment than insulin alone. I think insulin alone, to shorten it, to summarize, will be of interest, but not adequate as a risk profiler.
Thank you so much, Dr. Dannenberg. Due to the time constraint, we have to move to our next speaker, but of course we're going to provide, as I said at the end of the webinar, the email address for the speakers to send additional questions. So now I'm going to introduce our third speaker, Dr. Berger. Dr. Berger is the Hannah Payne Professor of Experimental Medicine, Professor of Medicine, Biochemistry, Genetics, and Oncology, and Director of the Center for Science, Health, and Society at Case Western Reserve University. He is also the Director of the Case Comprehensive Cancer Center Program on Aging Cancer at the Case Western Reserve University School of Medicine. Dr. Berger will present on the topic Obesity and Cancer, Gaps in Knowledge and Future Directions of Research. When he will finish, we will have time for a few more questions. Dr. Berger?
Thank you, and good afternoon. When we think about approaches to control obesity and its impact on cancer, we think about in two categories. On the top is obesity control, and down below is what do we have to do to disrupt the obesity-cancer linkage. In terms of obesity control, the primary goal is to prevent obesity, and that's mainly through lifestyle activities, either physical activity and diet. And then the secondary approach is weight reduction, and we have pharmacological options, and 3 of the drugs that have recently proved effective are listed here: Contrave, and Belvic, and Casima. When I say effective, we need to remember that they're associated with about 5 to 10% weight loss, and it's really difficult to maintain that weight loss. Another approach is bariatric surgery, and that's already been shown to prevent some primary incidence of tumors, but it's interesting that it occurs, the cancer prevention occurs primarily in women and not at all in men. Some of the obstacles to obesity control are listed here. So one is the fact that we all seem to be programmed, more or less genetically programmed for energy storage, and the programming, the differential programming, has been associated with polymorphisms in FOXO, with mutations or polymorphisms in uncoupling proteins that reduce energy loss and heat generation. We're also exposed to abundance of high energy dense foods, and most of them taste good. There's poor adherence to caloric restriction. Nobody wants to do it. We have inadequate sleep nowadays, with disruption of our diurnal rhythm, and there's a clear relation of obesity and increased cancer with shift workers. We have much more sedentary lifestyles, and there's been a proliferation of energy-saving devices, and increase in screen-based activities and simulated recreation activities. The built environment impedes our physical activity. We don't have to walk to the store. It's even hard to find sidewalks to walk on in many suburbs. Exercise equipment and exercise programs may be costly. I mentioned the fact that there are some pharmacological interventions, but they are small and probably not of long duration. And then there's the high cost and the consequences of bariatric surgery, with the best estimates now of bariatric surgery costing about $25,000 to $30,000 pre-op, and that doesn't include the follow-up. I think it's important to consider some of the gaps, to think about what some of the other postulated mechanisms are by which obesity promotes cancer, and that includes, number one, increased levels of bioavailability of growth factors like insulin and IGF-1, increased sex steroid hormones like estrogen, and the recent observation that elevated cholesterol levels in the right genotype can be converted to 27-hydroxycholesterol, which in itself stimulates the estrogen receptor. We have altered adipokines like increased leptin and decreased adiponectin, and a number of others, which I'll talk about. And as Dr. Dannenberg pointed out, the low-grade chronic inflammation and oxidative stress, which is associated with the metabolic syndrome and has been given the name of meta-inflammation. We also have altered microbiomes, in which changes in the intestinal microflora associated with obesity, especially increased firmicutes and decreased bacteroidetes, can potentially increase the availability of toxins, and the possibility that the altered microbiome and other aspects of obesity may affect epigenetics. And I think epigenetics, with obesity causing methylation of some tumor suppressor genes with obesity, and I should have said demethylation and increased methylation with exercise, and Paul Wade at NIH has recently shown chromatin rearrangements in obesity that simulate some of the changes that occur in colon cancer. We have a number of adipokines. These are peptide hormones that are secreted by or from adipocytes. Some of these are secreted from many other tissues, but everything, well, it started first with the first adipokine identified in 1994 was leptin, which is the satiety hormone. It suppresses appetite. It also stimulates tumor cells, and especially stem cells, and not only does it stimulate growth, but it also stimulates proliferation in cells that have the appropriate leptin receptors. I'm not going to go through each of the others, but each one of these has been shown to have effects both in weight regulation and regulation of diabetes, but each of them has been shown to have an impact on one or many types of cancer. When we initially started thinking about this situation, the concept was that excess weight caused adiposity, which led to increases in free fatty acid, changes in some adipokines, insulin resistance, which led to the secretion of more insulin. The insulin had several effects, including changing IGF binding proteins, which led to more IGF-1 synthesis, and the fact that both insulin could react with an insulin receptor and IGF-1 could react with a receptor, and that would stimulate target cells to decrease apoptosis, increase cell proliferation, and cause tumor development. That was the simple version. What was going on more recently, we understand that all these other factors can be interactive, stimulate each other, and cause all the different components associated with cancer cell promotion and progression. And I've shown you here that obesity stimulates these effects in adipose tissue, stimulates the aromatase, as Dr. Dannenberg discussed, stimulates estradiol, which can cause all these effects, but you can see that other factors like PAI-1 and VEGF can also interact with these pathways. And so there are a number of routes that obesity can take to stimulate cancer cell proliferation. I also want to point out that not only does obesity cause all these effects, but many of them are stimulated by dietary fats. And what I showed you before was on the outside of the cell. Now I'm showing you here what happens at the cell membrane. So each of those factors has a cell membrane receptor, and it can stimulate all these different pathways which interact, and all of which can lead to cell growth and tumor promotion and progression. And it's quite clear that if you block one, there's a good chance that one of the other pathways or that one of the other receptors may activate another pathway that will stimulate cell growth and tumor formation. And I mentioned just before that dietary fat can cause obesity, leading to inflammation and a number of these other factors. But in addition to the dietary fat causing obesity, a direct effect of dietary fat, mostly with saturated fats, will also lead to inflammation, whereas a number of other dietary fats, the so-called healthy fats like oleic acid, the omega-3 polyunsaturated fatty acids, will block the inflammation. And this has been amply demonstrated both in epidemiological studies and in some animal models to either stimulate cancer promotion or, in fact, to increase apoptosis and decrease cancer promotion. Can you advance the slide? Okay, so some of the other factors that affect energy balance in cancer are myokines. These are muscle-secreted polypeptides, and a number of these either cause inflammation in the muscles, reduce inflammation, may inhibit muscle growth. Irisin has been shown in white adipocytes to induce uncoupling and increase thermogenesis. Spark, which is secreted protein acid enriched in cysteine and otherwise known as osteonectin, has been shown to reduce growth of colon cancer cells in tissue culture. And I show you here a diagram which indicates some of the many myokines that are secreted in response to exercise, with particularly emphasis on this spark protein and another calprotectin, which has been shown to inhibit colon carcinogenesis, but a number of these others which have anti-inflammatory effects, and others which may either lead to or present muscle wasting in terms of cachexia. In addition to the adipokines and the myokines, we also have a host of gastrointestinal peptides, all of which have been shown to be active in some degree of modulating energy balance, appetite control, glucose, insulin homeostasis, obesity, and growth control. Again, I won't go through each of these, but each of them has been shown in one system or another to be associated with increase or with some sort of malignancy. You're probably most familiar with gastrin, which stimulates gastric acid secretion is commonly elevated in multiple aspects of multiple endocrine neoplasia and leads to duodenal and gastric ulcers and malignancies. I've listed here some of the basic gaps in knowledge by which obesity impacts cancer. So one of the key questions is whether obesity is actually a carcinogen or a promoter of tumor progression. If it's a carcinogen, it means we have to deal with it in an entirely different way than if we think it's a promoter. And I can tell you that at least in animal models, 99.9% of all animal models, obesity will not cause cancer unless there's a preliminary mutation present. And so that leads to the question, are cancer driver genes the same or different in lean and obese patients? And depending on the answer to that question, there's some strong implications about how you would treat cancer in an obese or a lean person. The question of does obesity interact with the microbiome to drive cancer? The question of what is the nature of the adipocyte interaction with the microenvironment and the tumor cells, and can the normal and tumor-promoting effects of some of the peptide hormones be separated for therapeutic advantage? So, for example, the B insulin receptor is decreased in breast cancer relative to normal tissue. The A insulin receptor, which is also the fetal form, is increased in tumors. And can we design insulin molecules that would take advantage of this difference? These are some of the gaps at the translational level. I'll simply say, when does obesity impact cancer? And we have all these possibilities. Again, are the mechanisms the same or different? Should interventions be the same or different at different time points? And I think we really need to decide for any experimental system, what are the goals of our intervention? Are we trying to get primary prevention? Do we want to improve compliance or therapeutic efficiency? There are a tremendous number of studies out there on treating patients who already have cancer, and the goal is to improve the quality of life. But are we interested in reducing recurrence and enhancing survival? I mean, I think the answer is clearly we are interested in these approaches, but these are really, really long experiments. So here's a summary of some of the obstacles to disrupting the obesity-cancer linkage. One is the multiplicity of obesity-driven extracellular hormones that I showed you. The other is the multiplicity of obesity-driven inflammatory factors. The third is the fact that the receptors can hybridize, crosstalk, and stimulate growth pathways. The fourth is the multiplicity and crosstalk of the intracellular pathways. The fifth point is the multiplicity of cellular targets. Are we really, in any intervention, we need to know what we're targeting: the cancer cells, the cancer stem cells, the tumor microenvironment, and also, at this point, the lack of clarity on timing and targets. Are we looking at mutagenesis, promotion, progression, or recurrence? So here's my suggestion, and I'll make the disclaimer that this is my suggestion, doesn't reflect anybody else's example of how you develop an intervention to disrupt obesity-cancer linkage. First, you need to select a process and a time period, so you might want to target epigenetics, and if you do, you might want to look at epigenetics in the prenatal time period or the adolescent period. You might want to look at cancer promotion. You may want to target the metabolic syndrome and meta-inflammation. Then you need to identify an intervention. The intervention, as Dr. You might want to prevent maternal obesity if you're looking at epigenetics. You might want to change the dietary component. You might need to increase physical activity. And we need to identify a biomarker. We do not have a good biomarker. We need the equivalent of a hemoglobin A1C, which is nobody would treat diabetes without following hemoglobin A1C, and we need a marker like that for obesity. Also, nobody would treat diabetes without multiple approaches, and so we probably need multiple approaches to this. And then you need to identify an endpoint, which I have listed several choices here. And this is just an example of saying if you wanted to disrupt the obesity cancer linkage in women treated for breast cancer, again, the first thing you do is select a process. Let's say we pick time period adults being treated for breast cancer and cancer promotion, metabolism, and meta-inflammation, and then you identify the intervention. And again, I think you need to make it comprehensive, so it probably requires diet change, pharmacologic intervention with weight loss agents, anti-inflammatory agents, and increasing physical activity. Again, as I said before, we really need to identify a biomarker, and the question is, is there some way we can get a local biomarker, or is there still a systemic biomarker out there? And then what's the endpoint? Do we want to prevent recurrence, increase survival? And I think on the one hand, the ultimate goal is to prevent cancer, but if we're treating obese patients with cancer, the most important thing that we want to do is prevent recurrence and increase survival. And then the fifth challenge is to figure out how do you, it's such a long process, how do you develop a randomized controlled study? And I think with that, I'll stop and thank you for staying with us, and I'll be happy to answer questions. Hello?
Thank you so much, Dr. Berger, for great presentation. We have received a number of questions for you, and I'll read the first question. Can you address dietary fat as a cause of obesity? Are you referring to a hypercaloric diet or any diet with a higher percentage of dietary fat?
So I think it's all of the above. I mean, I think people talk about the Western diet versus the Mediterranean diet, and depending on how rigidly you adhere to the Mediterranean diet, you're presumably going to have less weight. But nonetheless, we know, we're aware of plenty of pictures, at least, of people who are enjoying the Mediterranean diet and are just as obese as people on the Western diet. But nonetheless, a diet, high in fish oils and nuts and some of the other components of the Mediterranean diet seem to be less prone to some of the problems associated with the high saturated fat diets that we have in the Western diet.
Thank you. And the next question is, what anti-inflammatory would you recommend for including in a comprehensive randomized controlled trial in this population?
Yeah, so that's an outstanding question, and that's an important question that really needs to be answered. And I mean, I think that the best recommendation now would have to be based on the fact that we know that there's a lot of one of the major problems with stimulating cancer is involved with prostaglandin metabolism, and it interfering with some of the prostaglandins synthesis, it has shown some effect. So at this moment, I think the best thing that we could recommend is, or that should be tried, for starters, is aspirin, non-steroidal. But I think that we need to do a lot of work. That's one of the gaps in knowledge, so to speak, and one of the areas where a lot of work needs to be done.
And the last question is, if a foundation will fund a study of your choice, what would be the topic of that study that you would choose?
If a foundation would fund the topic of my choice?
Yes, what that topic would be.
Well, so I think two things. I think what's most important is to do a clinical trial, and we are doing several clinical trials, and it's interesting. We're doing trials in uterine cancer. We're doing trials in breast cancer. We're doing clinical trials in prostate cancer. And quite frankly, the one where we get the most cooperation is for women with breast cancer. And so based on that theory, it would be to do sort of a more definitive approach to that, and or for lots of other reasons, say to colon cancer.
Okay, thank you so much, Dr. Berger, and thank you, Dr. Giovannucci and Dr. Dannenberg, for your insightful presentation. Thank you, our audience, for participating today. I hope you found these presentations both inspiring and practical, and you will use the information toward your future work. Indeed, obesity is an important risk factor for cancer, and we learn more about some common pathways, some gap in knowledge, some mechanisms, and we encourage researchers to continue their work and help to answer the remaining questions. Nutritional Science Research Group in the Division of Cancer Prevention is eager to assist with your research. And to learn more about Nutritional Science Research Group, please visit us on the website that you see on the slide. Also, if you have future questions, please contact us at nutritionwebinar@mail.nih.gov. And also for information regarding clinical trials, please visit clinicaltrials.gov. Upon completion of this webinar, you will receive an email with information about obtaining continuing education credit. Thank you all for joining us today. Goodbye.
Thank you for joining today's conference call. The call has now concluded. You may disconnect. Thank you for your participation.